Have you ever wondered how thousands of chemical reactions happen inside our body every single second without taking hours? The answer is enzymes, the biological catalyst that makes life possible. In this video, I will explain enzyme nomenclature, classification, and the properties in simple way. MBBS exam oriented way. By the end of this session, you will understand how enzymes are named and six major classes of enzymes with examples and the important properties that are frequently asked in university exams. So, grab your notebook and let's make enzymes easy to understand. Let's get started. Definition of enzymes: biocatalyst synthesized by living cells and a substance that increases the velocity or rate of a chemical reaction without itself undergoing any change in overall process. These are the synthesized by living cells, hence it also called the biocatalyst. And catalyst, why? It increases the velocity of or the rate of the chemical reaction without itself undergoing any change and overall process. Then characteristics: protein in nature because they are formed by the peptides, but there is one exception, that is RNA. It is act as a ribosome. And then colloidal nature, thermolabile means the break after further level or specific level of temperature. It is specific in action, synthesized by living cells, that biocatalysts, also called as biocatalysts, and useful for disease diagnosis. We will see in further videos. Nomenclature and classification: International Union of Biochemistry IUB, based on their action, they are divided into six major classes. Each enzyme is assigned 4-digit code number. First number shows the enzyme class. First one is class, second figure shows subclass, and third figure shows subclass, and fourth figure is serial number of the enzyme in its subclass. See about the classification of the enzymes. Here is mnemonic for that: OTHLIL. Enzymes are divided into six major classes. Each class on its own represents the general types of reaction brought about by the enzymes of the class. First one is oxidoreductase. O for oxidoreductase, T for transferase, and further H for hydrolases, L for lyases, I for isomerase, and finally C for ligases, L for ligases, and here one new class, translocases. Oxidoreductases catalyzes oxidation and reduction, means electron transfer reactions. Here AH2 plus B, here hydrogen transferred to the B, and it occurs reduction, here oxidation. Hence it is catalyzed by the oxidoreductases. These are the example of alcohol dehydrogenase and NAD plus oxidoreductase. And here is the nomenclature for this enzyme: 1111. Transferases transfer functional groups between the molecules. Example: hexokinase, transaminases. And here A bond to the X is transferred to the B. Hydrolysis break bonds using water, called hydrolysis, and here are the example: lipases, cholinesterase, proteases, lactase. This is the reaction. Lyases remove groups to form double bonds or add groups across the double bonds. These are the examples: aldolase, decarboxylase, fumarase. Here addition and elimination is occur. Here addition and here elimination. And fifth one is the isomerases. Rearrange atoms re- within the molecule means the isomerization reaction. Phosphoglucomutase or the retinol isomerase, ketoisomerases. These are the examples. Sixth one is ligases. Join two molecules requiring energy, that are the synthetases, carboxylases, glutamine synthetase. This is the reaction. It's originally condensation reaction and usually ATP dependent reaction. Here is the one new class, that is the translocases, move molecules across the membranes, that is ATPases. Here we will see about the chemical nature and properties of the enzymes. Enzymes have these are the properties, that are enzyme sensitivity, enzyme specificity, enzyme catalysis, lower activation energy, reusable, and enzyme regulation, and enzyme specificity. Specificity means any specific reaction requires that specific enzyme only, not others. And then lower activation energy we'll see in further videos, and it is the reusable enzyme, can't waste after one use. It is the reusable enzymes. Let's see, functional unit of enzyme is also known as the holoenzyme, which often made up of apoenzyme, that is the protein part, and the coenzyme, that is the non-protein part. Here is the holoenzyme means the active enzyme that is formed by the apoenzyme and coenzyme. Apoenzyme is protein part and coenzyme is non-protein part. You can also remember protein part like P for P, apoenzyme having P letter, and coenzyme not have P letter, so non-protein part. Here just for remembering. Prosthetic group is used when non-protein moiety is tightly means covalently binds with apoenzyme. Coenzyme can be separated by dialysis from the enzyme, while the prosthetic group cannot be. Here coenzyme can be separated by dialysis method from the enzymes, but prosthetic groups cannot be because it is tightly binds with the apoenzymes. Oligomeric enzymes, which are the polypeptide chain, which have polypeptide chain, an example is lactate dehydrogenase. Monomeric enzymes have the single polypeptide, that is as an example ribonuclease, trypsin, and multi-enzyme complexes that are possessing specific sites to catalyze different reactions in sequence. Examples are pyruvate dehydrogenase and fatty acid synthase and prostaglandin synthase. Here we have covered the properties, chemical nature, and these are the classification nomenclature for the enzymes. If you like it, share, like, and subscribe. And in the next video, we will see next topic. Thank you.